Director of the National Centre for Clinical Research on Emerging Drugs and also the Clinical Director of the Alcohol and Drug Service at St Vincent's Hospital in Sydney. Before we get going, I just want to acknowledge the traditional owners of the land on which I'm seated today, the Gadigal people of the Eora Nation. Pay my respects to elders past, present and emerging, and any Aboriginal people who might be here with us today, and recognise that sovereignty was never ceded. Thanks for joining our 7th webinar in this series that was produced in response to COVID-19, and also want to thank 2 of our consortium partners in the National Centre for Clinical Research in Emerging Drugs, St Vincent's Health Australia and the National Drug and Alcohol Research Centre. It's no secret that we've been facing a whole lot of challenges during the COVID period and during the lockdown, and we wanted to explore some of those challenges and opportunities that have come during this time specifically for the drug and alcohol sector. And we've had a range of discussions with different people, consumers, healthcare providers, clinicians, through talking about some of the kind of skills and practices that we've been developing during this time. And today's guest, I'm really pleased to introduce, is Dr. Jessamine Soderstrom, and she's going to be talking to us a little bit about the drugs and the change in patterns of harmful drug use that have been presenting to the emergency department where she works in Perth. Jessamine is an emergency physician, a clinical toxicologist at Royal Perth Hospital, and also with the National Poisons Network. Forgive me, I'm going to read out Jess's bio because it's really quite exciting, so I want to share it with everybody. A self-professed nerd with a desire for a sedate life in an adrenaline-fueled environment, a desire to learn and a curiosity of mind, she's been privileged to work with an amazing team of researchers and clinicians to bring about a seed of an idea of an exciting project. It's the recognition and respect of each other's strengths and qualities that have enabled an environment where innovation can thrive. This journey was motivated by the burden and suffering seen by illicit drugs used in the community and a lack of information about what agents specifically were actually causing harm. We know that what people purchase may or may not be actually the substance that ends up being used, that we have no real way of telling what people are actually using apart from what it's marketed as, without any kind of good manufacturing process for the illicit market. So Jessamine was particularly interested in trying to find what those agents might be, and particularly as novel psychoactive substances are starting to appear in the market with at least some 500 different structures, and there was just no means or capacity to discover what agents were harmful. All presentations to the hospital would just have to be managed symptomatically, so it's, you know, how someone actually presents, whether they've got a fever or high blood pressure, and not actually knowing what the actual substance is. So this journey has opened doors to the benefits of collaboration, learning from others' expertise, and sharing of information with the aim to drive change in this area. The aim over the next few years will be to foster relationships between the Poison Centre, Sentinel hospitals and forensic laboratories all around the country, with a view to develop a national registry. EDNA, the Emerging Drug Network of Australia, will serve as a toxico surveillance network and the basis of the development of an early warning system for psychoactive drugs that are being used in the community. It's time to move beyond a band-aid treatment of the problem to being focused on reducing harm to people, including young people, at the peak of their lives. So I'm handing over now to Jess. Thank you for joining us. That's a great bio, so I don't know if there's anything more you want to add to that. Just before we start, if anyone wants to send in any questions, at the bottom of your screen there's a question button. Please feel free to ask anything that you'd like, and we'll answer them either as we're talking or at the end of the session. We've got about 25 minutes, so I'm going to hand over to you, Jess. Maybe you could start a little bit by explaining what changes you've seen recently.
Thank you, Nadine. So I work at Royal Perth Hospital, which is a inner city tertiary hospital in Perth. And we normally see probably about 85,000 presentations a year. And, you know, with COVID, we've seen a drop of about 25% of presentations in the initial period, although numbers have cranked back up to normal now. So we have been, so in April, which is about the start of the COVID-19 pandemic, what we have noticed was that, the number of presentations to the ED had dropped by about 25%. So we had hypothesized that we were anticipating that the illicit drug presentations would have likely dropped about the same percentage. And we've been doing sort of weekly snapshot audits in every April. That started last year, and we did it this year. So in 2019, we were seeing about 1,400 patients a day coming through the ED in a week. And during this time of COVID, the ED presentations dropped by about 25% to about 1,037 patients a day. Apologize by the door. And so we hypothesized that the numbers of ED presentations, that the equivalent would drop in the drug-related presentations. In fact, not. See, the anecdotal reports from the user groups were that drug availability had dropped with the border closures, with drugs becoming more expensive and perhaps less pure. It does not seem to have appeared to have deterred usage from our audit. In fact, there's been an increase in the number of drug-related presentations from 6.3 to 7.7% during that week. So pre-COVID, we were seeing around 8 a day.
Can I just
post-COVID, go
on? You mean that proportionally there was an increase, but the actual numbers, did they go up as well, or the numbers sort of stayed the same?
So proportionally, it actually increased. So total numbers have come down, but proportionally, it's actually increased. So we would normally see about 8 a day of methamphetamine-related toxicity, and during the COVID period, the number had only gone down to 7.
Yeah. Okay.
Does that make sense? So the total number of presentations of all ED presentations had dropped by 25%, but proportionally, the numbers of drug-related presentations had increased. Methamphetamines was still the most common drug used at about 62%. Interestingly, opioid use had reduced by about half, from 13 to about 6%. And clinically, there didn't seem to be much of a change in terms of the actual clinical presentations, with the same number of people, with them, the patients mainly coming in with, psychotic illness, and about 31% needing some form of sedation.
And how does these changes in the patterns of use? I mean, I'm particularly interested in the relative drop off in opiates, and the fact that methamphetamines have pretty much stayed the same. How does that feed into the EDNA project? And maybe you can tell us a little bit more about the EDNA project.
Yeah. So EDNA is the Emerging Drugs Network of Australia. The plan is that we're going to set up a national registry looking at what illicit drugs are causing presentations to an ED. So traditionally, when people come in with illicit drug poisonings, we would just treat them symptomatically. So if they were sleepy, we would wait for them to wake up. If they were agitated, we would give them some form of sedation, and we would have no idea what it is that they've actually taken. Now, this is quite different to how we manage pharmaceutical poisonings, where we would actually use drug levels to help guide treatment. And the role of this registry is twofold. One is as a toxical surveillance, and the second is as a basis for an early warning system. So we've been working with the forensic laboratories to develop a clinical protocol for the optimum method of blood collection and analysis to identify what drugs of abuse are causing symptoms and correlating that with clinical symptoms. We now have a sentinel hospital in each state that is linked to a forensic laboratory, who are all using the same clinical protocol for blood collection, and we are in the process of developing a database to store all of this information. So from our pilot project that we had been running for the last couple of years, what we had learned was that methamphetamines was by far the favorite drug in WA, with sort of comprising about 66% of presentations. Most of them came in very agitated, with at least 64% having some form of psychotic symptom. but 90% of them needed some form of sedation, with 70% needing about intravenous or IM, some sort of parenteral sedation. 2/3 of them needed security at some point, for physical restraint. Most people stayed in for about 11 hours, with most of them actually needing to stay in a main ED because it would just be behaviorally too challenging to be admitted to a ward. But what's really interesting is that from our preliminary data is that most people stayed for 11 hours and were discharged home, with only 2% of them having ongoing psychotic symptoms that needed admission to a psychiatric unit. And what was fascinating is that when we measured their meth levels to try and correlate it, and this is a cohort of people who are unwell, who are profoundly psychotic and agitated. When we measured their meth levels, and their meth levels was only half of that of police roadside sampling. So the median levels for the police sampling in WA was 0.21 milligrams per litre. In our cohort of patients, it was 0.11 milligrams per litre.
Is that surprising for you?
Sorry?
Is that surprising for you?
It was, absolutely. Because we had hypothesized that, our initial hypothesis when we started the project was that, people were coming in psychotic because of high meth. We assumed that it was because they were highly intoxicated with meth, and we would correlate it with high meth levels. Mm. And I'm going to make the bad assumption that if you're driving over for random sampling, that it shouldn't be too bad. And so the police samples, their levels were much higher than what we were getting. Mm. And that has been also, the police samples has been also been replicated in other countries, like in Germany. So we were quite surprised by that, because we had assumed that, people were coming intoxicated and psychotic because of high meth levels, which then had to make us think, perhaps it's actually not because of the high meth levels that is making them psychotic. Maybe it's because of the pattern of use. And it may be sleep deprivation that is causing the psychotic phenomenon. And especially with the fact that the majority of patients after 11 hours, their psychotic symptoms resolve. And the majority of patients, a small percentage having ongoing psychosis that needs psychiatric input.
Mm. And it also fits with what we know about tolerance to methamphetamine, that over time with chronic exposure to methamphetamine, people become tolerant to the stimulant effects, but actually lose tolerance to the psychotic effects. Mm. So it would fit also with that observation that you're seeing as well.
Yeah, but it also fits into the into a change of practice where with drug-induced psychosis, for us it was very reasonable to keep them in the ED or even ED observation ward for a period of time, allow them a period of sleep, and the majority of them will, the psychosis will resolve. And that just takes a bit of pressure off the psychiatric services. Mm-hmm. Rather than, all of these patients having to go to the psychiatric unit, that we can actually hang on to them because the majority of them will settle. Mm. Yeah.
And does that provide time also to link people in with ongoing treatment if they're looking for treatment for methamphetamine use disorders, or is that not something that can
Absolutely. So most of our patients, we would admit to our ED observation ward, and we've got a little section of the ED observation ward that's a much higher acuity ward, where they've got much higher nurses to patient ratio, and they are more they're capable of looking at very behaviorally challenged patients. So where was I going with that? So, and we do have a very close working relationship with psychiatrists, with social workers, with drug and alcohol that will come and see patients, before they get discharged and offer them. And we see it as an opportunity to link them into services and at least, plant the seed of whether or not they, plant the seed for them as to whether or not their drug use may be an issue, and if they want if they're ready to look for help.
Mhm.
Yeah,
Yeah.
so that's well established. Yeah.
And what are some of the more novel psychoactive substances that you've detected?
So methamphetamines is clearly about 2/3 of all the samples that we're getting. Of the other third, it's a selection of different novel psychoactives. So MDMA is probably the next most common. And we had a cluster of the synthetic hallucinogen N-bomb that was combined with 5-fluoroamphetamines. That particular cluster in about 2016, there was a lot of media out there about a big batch of bad MDMA causing a lot of deaths. But when you actually analyzed it, it wasn't MDMA at all. It was actually a combination of N-bomb and 5-fluoroamphetamines. And what was very interesting during that cluster was that what was reported in the news and in the coronial reports, with the 7 deaths in Victoria, 1 in WA, and I think there was a couple in New South Wales and in Queensland, that was only the tip of the iceberg. We had just in one AD, we had 16 presentations of NBOME and 5-fluoroamphetamines, of which they were very, very unwell, needing intensive care, but they survived. And we haven't seen that since. Yeah, and we haven't seen much of that combination of NBOME and 5-fluoroamphetamine since, which is quite interesting. Now, about the time when we started the project, the new legislations around the catch-all legislations around novel psychoactives came in. So we were expecting to see a lot of synthetic cannabinoids, but we hadn't seen any, hardly any, maybe 2. And it was mainly in the Fibonacci class. In the last year since we've been starting the EDNA project, we are starting to see an increased number of synthetic cannabinoids. And they're different cannabinoids to what we were seeing before.
So these are people actually presenting with acute problems. What kind of problems were they getting with the cannabinoids?
So they were coming in with agitation, psychotic, with psychotic symptoms. So that they presented looking like and so we were seeing, and what the types of synthetic cannabinoids are quite different. So we were seeing Kumal Pinaka, Cyanokumal Bitunaka, and Pegaklone, Kumal Pegaklone. So quite different compounds to what we were seeing a couple of years ago. And definitely a lot more in the last year than what we had seen for the last 2 years. So, and other sort of novel psychoactives we were seeing were LSDs, cathinones, PMA, GHB. And I think it sort of brings on, about how important it is to, in terms of, the toxico surveillance of this project, because what we were seeing was the pattern of drug use was changing. So with synthetics, and now we're seeing a rise in synthetic cannabinoid presentations to RED. And with the EDNA project, we've extended the synthetic from just looking for stimulants to also looking for opioid presentations. Because anecdotally, what we were seeing is that people coming in with opioid poisonings, we were needing to give much bigger doses of naloxone, and most of them actually need to go on naloxone infusions. So the question for us was that, was this heroin? Because it didn't really quite fit with the heroin presentations from when I was a young registrar, where you give them, a small dose of naloxone, and they often don't need more than one, and then they'll wake up and leave. So it was quite a different pattern, where what we were seeing was that people were coming with opioid poisonings and they were needing naloxone infusions. So of the patients that we have analyzed their bloods for, we've only found 4 patients with confirmed 6-MAM. And there were a few others with a morphine coating, but we're just trying to work with the lab now to determine what the ratio is to work out whether or not it was heroin or a synthetic coating. And in fact, the majority of the patients were, what we were finding, they were actually pharmaceutical opioids. So the pentodols, the oxycodones. So, and I think that's quite important from a public health perspective, and it makes sense as to why it is that the patients were coming in needing prolonged sort of naloxone infusions rather than, because I'm just wondering whether or not it's primarily heroin or it actually diverted pharmaceuticals. And the beauty about this project is that we are starting to see patterns around the country that are different. So in Sydney, they with the PRIZE project, they have been identifying some synthetic fentanyls with the acetyl fentanyls. And the lovely thing is that, we're sharing that information, but we haven't seen any in WA as yet. So there's been no synthetic fentanyls in WA. And clearly this is a big problem, overseas. So, so that sort of collaboration between the states and seeing what the patterns of use are, I think that's, incredibly valuable.
And is that something that is already established, or is that something that is part of the EDNA project, or what stage is all of that at?
Yeah, well, it didn't exist before.
Yeah.
So, besides the anecdotes and the, talking to our colleagues overseas, it goes, there was no notification previously. Besides, a colleague might send you an email saying, by the way, are you seeing some of this? And that's about the extent of the collaboration. Whereas now there is more of a formal link between the states between the states where we are notifying each other about what we're finding. As an example, there was a recent outbreak of atizolam that's sold as fake alprazolam. Now, this is an unusual novel benzo that has got a side effect of hypoglycemia. Now, we weren't sure initially whether or not it was just a one-off, but in fact, they were seeing it in New South Wales and we're seeing it in WA. And so we know that, and that sharing of information means that, together we've got a bit of a pool of information rather than just being the isolated one or two, the power of actually combining that information. Yeah.
So how does that fit in with the EDNA project?
They are a part of the EDNA project. So the PRIZE project in New South Wales, their clinical protocol and what they're doing is very much, the protocol that we've developed here. And they are also going to be putting in their data into the EDNA database. So where we're at now, in as far as the EDNA database goes, we've got a national steering committee that has a representative from the National Poisons Network and an ED physician, clinical toxicologist, and intensivist from a sentinel hospital in each state. That's linked to a forensic scientist and a data manager state data set. This is what we're going to collect nationally. We've got ethics approval for the national registry, and which will be a federation of state-based registries. The forensic laboratories through its national body, the TOXAG, are agreeing on a standardized library and a classification for the NPSs. So that's kind of where we're up to, and so the discussions are being had, and each state, and what we're going to do is that the database is going to be based on REDCap, which is fairly, which is available throughout Australia, and it will be hosted at Curtin University, who has got experience in terms of managing national registry.
And how does EDNA fit in with the prompt response network? I'm not sure everyone listening is aware of the prompt response network. Maybe you want to explain a little bit how you see that fitting in with the prompt response network?
Yeah. So part of the role of the registry is also to develop. It acts as a basis for the forming of an early warning system. Where what we realized was, as we were collecting this information, is that when we do get clusters of drug poisonings, that there was clearly a need for a systematic way of collecting that information and disseminating it in a systematic fashion. And kind of key components of that is that we needed a filtering system to actually confirm the information. And so in the case of WA, that's our Mental Health Commission, who will then decide whether or not the information would then be disseminated to just first-line responders, or whether or not that's ambulance, police, or the EDs, or whether or not there needed to be a broader public health messaging. The important thing is that our aim had to be to minimize harm. And this was very new to me, because clearly I'm an ED physician and I have very little experience in as far as public health messaging. That concept of chasing a red dragon, and the concept of chasing a red dragon and actually causing more harm and more use. We had a situation a few months ago where there were a few deaths that were attributed to GHB in WA. But none of those deaths were actually due to GHB. One of them was a sympathomimetic toxicity, and the other two look more like opiates. And we don't see a lot of GHB in Perth. And so we had hardly any presentations that weekend when these 3 deaths occurred. But the information went out that there was a big media output saying, oh, this is all this GHB deaths out there. And the consequence of that was that the following weekend, we had a big spike. From not seeing hardly any at all, we had 10 presentations the following weekend from GHB poisonings. And it just kind of demonstrates how important it is to ensure that, number one, we've got the clinical information correct, that the poisoning is because of the particular agent. And the second thing is, working out the best way to put out that information, whether or not it's just to inform the first line responders and the user groups, and, when we actually decide to put out a bigger public health message.
What are you suggesting that happened then?
Sorry?
Are you suggesting that people were nervous when they took GHB and they, out of caution, went to hospital, or?
No. There was just more use. There was a lot more use of GHB. Yeah. More harm present. More harm present. Yeah. Because people don't generally come to the hospital unless they are, unless they're unwell from the use.
So, I mean, the EDNA project, NCRED's been involved in, because I think you were successful in one of our seed funding rounds. So it's
absolutely
really for me to see this kind of take legs and get into something bigger. And it's really exciting for me to see it fit in with the prompt response network, which is exactly what you've been talking about, having a national level communication that also brings in consumers. So not only health providers, but also people who are using drugs and experiencing harm. So it'd be really great to see how this all kind of
the magical thing is, and I have to say thank you to NCRED, because this was just an idea, when we saw the grant and we applied for it thinking, okay, if we get the grant, then we can really get this going. And what has happened is, so what came up as an idea, our little pilot project, which was the WISE project, thinking this is a really good thing to be doing. We then applied for the grant, and now we've got a project officer that is absolutely pivotal in trying to drive it. And from that seed funding, we have now gotten the interest of the health department and the mental health commission, because they actually see you of the work, and they are, and we are just about getting them on board to actually help fund the project and help really drive the registry. And how this fits into the prompt response network is that I see ourselves as a bit of a piece of a jigsaw. So we are the ED front of drug use. And so what we can do is to tell you what the pattern of poisoning is like, what people are coming in crook with. And, so things like, are what people think that they're taking, is that what they're actually taking? What are the biggest drugs that are causing presentations to an ED? What is their pattern of poisoning? And moving away from anecdotes. Before people, we would go, oh yeah, we had a cluster of people saying they've taken some pink champagne. I'll never see pink champagne the same again. And it was just, a form of meth that they were using. So from that seed funding, it's now growing, and it's got momentum. So, I can actually, and Victoria have now, looks like they're going to get funding to get their project up and running. So it's very exciting.
Right, fantastic. Thanks so much for sharing with us today, Jessamine. And really, this is the start of a national collaboration. So look forward to checking back in a few months' time. Yeah,
and people are talking, and are sharing in a way that, well, I'm very naive in all of this. I think I've got information, I want to share it, and and other people, when they rather than just sort of being very cautious about all of that information, they were going, you know what, this is our aim, is to minimize harm. And so broadly, if that's our aim, what do we need to do to stop young people from dying and coming to harm?
Excellent. All right, thanks, Jess. Thank you. We'll catch up soon. Bye.
Okay. Thank you.